Hello everyone, my name is María Fernanda Arredondo Vivas, and today I'm going to present to you an article called Berberine alleviates respiratory syncytial virus induced pediatric bronchiolitis and fibrosis via suppressing the HNGB1, TLR4, and NFKB pathway. So first, what is the RSV? RSV is the respiratory syncytial virus that is a common RNA virus that affects children and babies. Then we have our signaling pathway. So when the cells get infected with this virus, they release HNGB1 that binds to the receptor TLR4 in the nearby cells. When this occurs, the cells start activating the transcription factor NFKB that will induce some inflammatory cytokines. Now we have bronchiolitis, that is an infection in the lungs that affects especially young infants and children and will cause them swelling and mucus built up in the bronchioles. And finally, we have fibrosis, that is an excessive accumulation of fibrosis connective tissues in the lungs. Now we have our general objective. In this study, they wanted to explore the Berberine therapeutic potential and the molecular mechanisms in the RSV-induced bronchiolitis. Now with the methods. First, we have the quantitative reverse transcription polymerase chain reaction, in where they wanted to measure the expression of some genes. So what they do is that they extract the RNA, then they convert that RNA to DNA, because with the DNA is where the PCR works. Then this DNA is amplified and is the same with a fluorescence signal. So the more fluorescence it shows, the more this active gene is. So they wanted to measure the activity of RSV, HNGB1, cytokines, and the inflammatory and fibrosis markers. Then we have the flow cytometry. In the flow cytometry, they want to measure the state of the cells. So if the cells were healthy, were going under pyroptosis, or were apoptotic cells. So they stain those cells with an enzym B, and that will dye them if they are apoptotic, or with propylium iodide if the cells are going under pyroptosis. So they put these samples in a laser passed through it, and it will show some fluorescence. They wanted to measure the effects of the RSV in the infected cells and if the Berberine could reduce it. And also they wanted to see if the overexpression of the HNGB1 could affect the Berberine effects. Then we are going to have the Western blot. With the Western blot, they want to measure the amount of a protein in a sample. So what they do is that they separate the protein with gel electrophoresis, then they mark that protein with an antibody, and this will make a chemical reaction that will show a band. So the bigger and the darker that band is, the more that protein is expressing. And they wanted to measure the expression of all the signaling pathway, the inflammasomes, and the fibrosis markers. And then we are going to have the enzyme link immune absorbent assay, where they wanted to measure also the proteins in a liquid. So what they do is that they use two antibodies, and one of them is linked with an enzyme that will make that protein change its color. And this was specifically used to measure the amount of inflammation and cytokines. So let's start with the results. In this result, we have, in all of them, we have some Western blots. In this case, we are going to have here that it will show the expression of the HNGB1, and this is just a control. So in the blank cells, as you can see, the HNGB1 is not expressing, but when they infect the cells with the RSV, it's going to start expressing. And here you can see when they use the Berberine. Then we are going to have here also the expression of the HNGB1, and they want to see here if it was correctly overexpressing. So here in the blank cells, as you can see, the HNGB1 is not expressing, but when they overexpress it in the blank cell, it will express. Now here they wanted to measure some pyroptosis proteins. So as you can see, here is our blank cell that will not express this protein. Then they infect the cells with the RSV, and they will start expressing those pyroptosis proteins. But when they start using Berberine, you can see a reduced effect. And here we had some fibrosis and inflammation markers. So in the blank cell, as you can see, it's not expressing. Then when they use the RSV, it will start expressing. And when they overexpress, the HNGB1 is going to block the effects of the Berberine. But when they block the receptor of the HNGB1, then those effects could be reduced when using the Berberine. Here we have a flow cytometry. So here we have our blank cell. This will show up how many cells were alive. But if the more amount of cells is here, then we have more apoptotic cells. As you can see, when we have the RSV, there are still some healthy cells, but most of them migrate to the apoptotic side. And when they start using the berberine, they could reduce that effect. Here we have some fibrosis and inflammation markers that will not express in the blank cells, will express in the RSV, and will reduce their effect when they block the receptor. And here we have an immunofluorescence that will show the expression of the COL1A1, that is a fibrosis marker. In the blank cells, it will not express, but in the RSV cells, it will have a bigger expression. When they block the receptor of the TLR4, they will reduce its effects. And finally, here we have the discussion where I mentioned 3 authors that all agree with what the article author said over the berberine effects and also of the RSV induced the signaling pathway. So to conclude, we have that the molecular biology tools were essential to understand how the RSV induced inflammation, pyroptosis, and also fibrosis. But how can the berberine reduce those effects? This cannot be seen with normal clinical methods. And also we have that the molecular biology is essential to start working on new therapies such as the berberine when we can identify specifically certain ways that can be targeted to reduce the tissue damage. Thank you.